the panel today is going to talk about more challenges that we see with irAEs, and to help direct us in the next 3 to 5 years of where they may see solutions. And again, we have panel members from various backgrounds to help us develop this. So to introduce Elad Sharon, he's a senior investigator with National Cancer Institute. Shaily Arora, she's the acting assistant director for safety in the Oncology Center of Excellence with the FDA. Mark Anderson, who's interim director at UCSF Diabetes Center. And last, Trang, a safety physician with EMD Serano. So we're going to immediately get into the discussion. And I'm going to ask each of you, first of all, what significant irAE challenges do you see from your perspective that maybe already working on and maybe that's not even identified yet? So I'm going to go to Elad first.
No, thanks so much, Andy. This is a wonderful group of really highly distinguished folks, and I'm honored to actually be a part of it, and I'm really proud of the work that we see at Roundtable and Project Data Sharing has been involved with up until now and continuing for the future. As you know, the NCI is actually very interested in this area and really trying to see what we might be able to do to help accelerate the improved understanding of immune-related adverse events using tools at our disposal. So one of those tools that I think that actually could work very well in coordination with efforts such as these happens to be the immune-related adverse event biorepository that we have begun working on back, I believe actually in 2016, 2017, when we initiated the work, setting that up with the Alliance for Clinical Trials in Oncology. And the goal in that particular instance is to utilize patient samples and collect data from patients that are being treated with immune checkpoint inhibitors and frankly, even other immunotherapies. We're at least able to take care of other immunotherapies as time goes on and more get approved. But taking samples from those patients and getting to the point where we might be able to learn that much more from these adverse events as they occur in real time. We're all aware that there are going to be some patients that we treat with these highly innovative therapies that end up having poor events or poor outcomes. And, we heard one of these stories actually today, and they're all actually quite unfortunate. Even ones that don't necessarily result in a patient's death also allow for us to have an opportunity to really intervene on a patient's behalf if we actually knew the right way to do it. And so in that sense, what we're missing is not just an issue of categorization, which I think the Delphi process and the neurologic and dermatologic teams are exhibiting extremely well, but also the ability to do basic and translational research with samples that are obtained from patients who are exhibiting these particular types of symptoms. And again, in order to do that, we need to actually be organized. We need to have a framework to make that happen. And the next step, beyond the question of what to do from the sample collection and data collection standpoint is, frankly, maybe I'm just biased because I'm a clinical trialist, but I think the answer at that point is to start working on clinical trials. And we've already gotten some, what I would call baby steps in that direction from a variety of different investigators who've suggested things to us. There's one effort that's ongoing that's actually sponsored by the NCI, led by Jarushka Naidu, is currently at the Royal College of Surgeons, actually in Dublin, but previously at Johns Hopkins, and she was working with ECOG Akron in developing a protocol for patients that had steroid refractory pneumonitis. The problem with an effort like that is that those types of efforts necessarily rely on these rare patients that you might not really see at one individual center. Even on a multi-center basis, they might actually be quite rare. And so I think that if you're asking at least me what I think we should be doing and what we can be doing into the future, my suggestion, and I think others have stated this as well, is that a multi-center, multi-toxicity trial is an important aspect of our work going forward, in the sense that any one center might actually have enough immune-related adverse events on their own, but not necessarily the tailored treatment that one might want, let's say, just for pneumonitis, just from their one institution. But all the institutions that are academic and interested in these particular topics, also community oncology centers that are interested from an academic perspective in developing better therapies for the patient, could potentially be contributing patients and contributing knowledge and know-how so that you can get to that next stage for patients going forward. And we certainly need these efforts. We need better categorization. We need better treatment opportunities. We need the data and sample repositories that I mentioned. And we probably need to do more high throughput, high patient number, mass analysis to determine if there are, in fact, particular autoantibodies that we might be able to find that predispose a patient, microbiome subsets that make a patient potentially more susceptible if they happen to have certain species of bacteria or a prevalence in their gut or elsewhere in their body, HLA subtypes. These are all things that we could be doing if we were organized and paid attention to the dual use that we really have from some of the efforts in oncology. In other words, the work that we're doing to determine what is immunotherapy resistance or what might lead to immunotherapy response is directly relevant to all these efforts as well. And all of it is really just meant to help the patient. So we need to be better organized and to work better together.
Thank you, Elad. So I'm going to switch it up a little bit. I'm not going to go in the order that I introduce everybody. I'm going to leave the Shaily to the last, so she doesn't taint the group here. But I'm going to move to Mark Anderson. What challenges do you think are there, and just go ahead with some of the solutions that we may need to address.
Yeah, so just as a way of introduction, I'm a physician scientist at UCSF, and I run a laboratory that's focused on mechanisms of immune tolerance and autoimmunity with a particular focus on type 1 diabetes. Mm-hmm. And so I have perhaps a different lens than some of the other people that may be in this community that's gathering this morning. So I had become aware of checkpoint blockade literally through my clinical activity as an adult endocrinologist. One of our fellows called me several years ago and saw a patient that had just gotten anti-PD-1 and developed something that looked like type 1 diabetes acutely, and asked me if I was interested in this. And I said, absolutely. And this led to me getting into the field and seeing this as a huge opportunity for our ability to understand why type 1 diabetes may happen, but of course also why is it happening in these particular patients? How can we come up with better ways of predicting someone that's going to get this particular complication versus another complication? And of course, because I'm an endocrinologist, this has also piqued my interest in other endocrine complications that commonly occur under the coverage of checkpoint inhibitor treatment that include hypophysitis and thyroiditis. And so one of the hopeful things that I'll just bring up that I saw this morning was the incredible network that the group at MGH has put together that's collaborative. And I think that this is really, really important. The clinical groups getting teams together, recognizing that there are experts that can bring something to the table I think is extremely important. And, what I have seen overall over the last 4 or 5 years of being involved in this research is that that's actually been a major impediment, is breaking down barriers. I see the cancer physicians frequently trying to keep everything to themselves, particularly when it comes to the research aspect of this, instead of reaching out to the colleagues that have expertise on the various complication diseases that happen in this situation, and working with them collaboratively. So there's a huge, social experiment going on with all of this. It really is important to collaborate. And then I think, as Elad just said, that many of these complications are quite rare. The one that I'm interested in, autoimmune diabetes, probably only happens in 1% to 2% of all comers that are getting checkpoint blockade treatment. And so it really forces you to collaborate with other groups if you're really going to seriously study this, where you can get enough patient material and samples to start seeing some signals. So then I think another challenge that I think has come up over and over again is heterogeneity in defining what disease actually is. And we're no exception to that challenge. When you're looking at cancer patients that are getting treated here, they're going to be in an older group of patients. Type 2 diabetes is going to be a very common feature of many of these patients. How do you know this is just an exacerbation of underlying type 2 diabetes versus it really is autoimmune diabetes? An interesting phenomenon that we've run into with type 1 diabetes that many of you know is the autoantibodies in general for garden variety type 1 diabetes are actually pretty good. Over 90% of patients that present with garden variety type 1 diabetes have one of our established autoantibody markers. Well, with checkpoint inhibitor induced autoimmune diabetes, it's more on the ballpark of 40%. So those other 60% of patients that are presenting, they don't have the autoantibodies. We don't really have a clinching diagnostic marker to say this is autoimmune. And then layered onto it is just another challenge I put forward for us that really is an impediment, is path material from the pancreas. We can't see what's going on in the islets, and this has been something that's been vexing the field even before checkpoint inhibitor treatment had come along. We really don't have the ability to see what's going on in the islets as far as the autoimmune response goes. And so figuring out how to work in a collaborative network where we could get access to such tissues, perhaps in patients that end up having a poor clinical course. Perhaps there could be organ donation where we could see what was actually going on in their pancreas, and doing that in a collaborative fashion with their colleagues in pathology is very important, and getting those people on board to understand that the importance of this, and of course the patient families to understand this. So what I see is there are many moving parts. And then another important challenge to bring up is there's just many events going on in these patients. Their immune system is attacking their cancer. Now their immune system is attacking their skin. Now it's creating an inflammatory process in their gut or their lung. And then you're measuring things that are going on in their peripheral blood. How do you know what you're measuring has anything to do with one or all or some of those features in breaking that apart? So large sample numbers, again, I think are critical. One other thing I'll bring up that I think is particularly interesting to our field in endocrinology is you'll see over and over again in irAE summary slides is this condition called hypophysitis, which is an autoimmune attack of the pituitary in patients. And what I will tell you is spontaneous hypophysitis is a disease in endocrinology. For the average endocrinologist at a tertiary quaternary care facility, it's maybe twice in a career disease to see. And now it's happening frequently in these patients that are getting immune checkpoint inhibitor treatment. And because it's been a very, very rare disease, not much is known about it. There aren't any autoantibodies. There's not much path material that's out there. Nobody completely understands exactly what the mechanisms are, other than it's likely to be autoimmune in nature. And so just understanding that alone, I think is very important. Again, it highlights the importance of the endocrinologists collaborating with the oncologists, collaborating with the immunologists that can look at these mechanisms in greater detail. But overall, what I see is a community that's building that I think is particularly exciting, because together I think we really could use this not only an opportunity to help the patients that are getting treated in a more precise way, help them prevent getting their complications, but imagine helping the patients that are having autoimmune conditions where they just had no understanding of why this is happening. And now, we've clearly established the fact that checkpoint inhibitor treatment patients are developing type 1 diabetes. The community of people out there that have type 1 diabetes and are advocating for type 1 diabetes, they see the enormous importance of this as perhaps an opportunity to understand how immune tolerance is being controlled to prevent the attack of the islets, and perhaps this whole disease entity is an opportunity to come up with new ways of thinking about treating type 1 diabetes. And so I think it's not only this community, but the community of patients out there with autoimmunity where we need the dialogues to also emerge.
Thank you, Mark. Yeah, I know as an internist, going back to the dermatological, I would have just put rash down. That's all. That's as good as I would get too. So.
Maculopapular.
That'd be a stretch for me. So thank you. So Key, as you look at this from a patient safety perspective and the safety physician at EMD Serano, what are some of the challenges that you still see, as well as some of the solutions you'd like to see implemented over the next few years?
Yeah, many thanks also from my end for this symposium, and it's really great to see how much work all the colleagues have put into this topic already, and it's really great to see how far they are. Indeed, the main challenge, what we see is really, and this is very much in line to what Dr. Guidon or Burton, as well as Dr. Chen and Louv had said with regard to the right diagnosis, right? So the point is indeed that due to perhaps the lack of specific definitions, what we really see is that often only very unspecific reports about any adverse events are just reported with the consequence that really a very clear identification as well as a good diagnosis of an immune-related adverse event is missing, which makes it more difficult, to identify these diseases and these reactions. The point is these unclear diagnoses in that extent are not very helpful for the patients in the end, because of the fact that the better the right diagnosis can be found, the better ultimately the patients could be treated, and the more specific the patients could be treated, and the more correct in the end the patients could be treated, right? So these are in essence indeed, and I'm really very happy that all the colleagues had mentioned this. I think it's really about the right diagnosis and the right identification of the disease entity. And having said that, I think in order to overcome this, I think it's indeed those activities that has been started here, I think fostering the understanding of these immune-related adverse events per se, but also sharing these informations, as you correctly mentioned before. I think those things are really the solutions that we need in order to really get a better understanding, not only from this panel, right? I think everyone here in this symposium knows the immune-related adverse events very well, but I think it's also every physician at every patient's bedside who should somehow keep this in the back of their head when treating a physician who is using an immune checkpoint inhibitor, that something like an immune-related adverse event could occur and could be observed. And I think with that said, the earlier such immune-related adverse events could be identified, for sure the earlier the patients could be treated more specifically and also much better. So I think, in essence, all what these initiatives are doing, I think, will definitely help in overcoming these problems that we are currently facing.
Thank you very much. Shaily, from your perspective, again, some of the challenges that you're seeing, as well as any solutions that you may have thought of, that we should be undertaking.
I'll start by describing a few challenges that we see from a regulatory perspective, and then hopefully talk a little bit about the potential solution. So even with the growing use of agents and increasing number of clinical trials, I do think that we are somewhat in the learning stages, and critical questions surrounding the identification, the characterization, and outcomes of irAEs are still remain to be answered. And as more and more checkpoint inhibitors are being used as part of combination therapy, I think it is becoming more critical than ever to better identify and capture the characteristics and severity and how they may have an effect on overall risk-benefit relationship. So talking from a regulatory perspective, pre-market standardization continues to be an area in need of development. And it has been addressed in earlier sessions today that there are unclear definitions of irAEs, as well as unclear diagnostic workups, which can lead to variability in identification of irAEs and what constitutes an irAE. So at the FDA, we see disparities across various applicants or BLA holders for describing the same medical concept. So we see disparate reporting of incidents and the characterization, which makes it really difficult for us to communicate it succinctly to our prescribers and our patients on how to manage these, especially for some of the common irAEs that are seen across the drug class. The inconsistencies also present challenges in undertaking research endeavors when we want to aggregate data across trials. So we want to make sure that we are comparing apples to apples. And I know all the caveats of having cross-trial comparison, but, for research and exploratory purposes, FDA has been trying, because we have this treasure trove of data of all these immune checkpoint inhibitors and the data that has been gathered in so many trials. And we have been trying to look and see if there are trends that we can analyze, and we have published some of our research. So I think harmonization of definitions and diagnostic workup, those are very important for us to create a stepping stone for doing future analysis. And the efforts that have been described in the symposium earlier are definitely paving the way, and PDS is playing a great role in moving the field forward. Some of the solutions that I would like to talk about, and I would like to begin by stating that we are getting better, but there is certainly a lot of work that remains to be done. And one of the crucial things I think that we, from clinical trials and regulatory perspective, would be to have consistency in data collection. So creating something like a common CRF, agreeing on common data elements such as which describe the symptoms, the duration of steroid use, where the need to re-escalate steroids, and also the timing of onset, because there is no defined way of how this is captured. Some define it as the time the steroids were started, but the symptoms could be appearing before then, and we know that low-grade irAEs are not well captured. So those are all the challenges that we see. But one of the initiatives that FDA undertook last year was to harmonize the labeling for all the PD-1, PD-L1 drugs, so that we have a uniform approach across the labels to provide risk mitigation strategies and provide information regarding dosing modifications and warning and precautions. And that was just an effort so that we are not giving different recommendations for similar or common irAEs, since they come from the same pathophysiological pathway. Another thing I would say that as more and more developments are happening in the field of immunotherapy, we are seeing research being done where 2 or more immunotherapies are being combined. And so it becomes more common, more important to have those common data elements and CRFs, and have to have that granularity in data collection and capturing of symptoms linked to interventions, linked to outcomes, so that we can really develop a data-driven risk mitigation approach, especially for those unique toxicities that Mark was describing, or some of that Elad shared, that those are not going to be seen in every trial with every patient, that they are unique and rare, and sometimes they require additional investigations. And FDA has continued to partner with PDS and other colleagues in pharma and academia to better understand these unique and rare toxicities, and so that we can have earlier interaction with broader stakeholders and come up with, as a group, on how do we manage these unique and rare toxicities. One other thing I would like to mention is that immuno-oncology is a key focus area of FDA, and the agency is really interested in the efforts to develop technologies and approaches, and some of the ones that Elad shared about the translational research. And because the ultimate goal would be to have, a greater understanding of doing predictive modeling, and not only to predict who would benefit from this therapy, but also understand these toxicities and come up with biomarkers to predict the severity and who will have experienced that, so that we can tailor the management as well as some of the mitigation approaches. So those were some of the solutions that I would like to state. But one common thing that FDA has been very vocal about is about expanding the eligibility criteria, making our patients in clinical trial more representative of the patients that are actually receiving these drugs in the real world setting, so that the data that is coming regarding safety and tolerability is more representative. FDA has also been very vocal about including PRO assessments as part of clinical trials, and has also published about recommendations and guidances, and has put up annual workshops. So I think those areas continue to be challenging, irrespective of irAEs. But once we have those incorporated, I think we will have a much better understanding of safety and tolerability profile. And as we move more and more towards using decentralized clinical trials, I think it will be good to have all these elements aligned and harmonized, so that the data that we gather from the decentralized clinical trials can actually be used for regulatory purposes. So that's what I would like to add.
Thank you, Shaily. And you're right, the decentralized clinical trials will add a new complexity to everything going forward. So, I want to thank all the panel members for sharing their insights. And thank goodness it's recorded, so we can kind of go back and look at this again. But I see a lot of opportunity in really helping the patients in the long run, and making sure they can continue therapy to treat the cancer, as well as ameliorate any risk that they may have adverse events. But I also see this as a great opportunity. I mean, when I was in practice, someone would come to me with an inflammatory condition. It was already going. This is a great opportunity to get these biospecimens before somebody's even started on these checkpoint inhibitors, before the immune system has been turned on. And really, I see this as an opportunity to help other diseases beyond those with cancer too. So this is really, I think it's very important, not only to immuno-oncology and, cancer therapy all along, but really to other diseases in the long run. So I want to thank Key, Mark, Shaily, and Elad for sharing your insights. Now I'm going to turn things over to Bill Louv, who's the president of Project DataSphere, to give us some closing comments, and thank everyone for attending today. Bill?
Thanks, Andy. I'll start by just thanking everybody. The contributions were uniformly great. Really appreciated all of them. Just a couple of thoughts to close out the meeting. Kerry Reynolds mentioned the Project DataSphere or FDA Project DataSphere Symposium 7 on irAE, which was 2 years ago. And in the call to action segment of the symposium, improving definition of the toxicities was identified as a core requirement to make progress. And today is an update on how we've made progress against that call to action. And I think you can see that Kerry Reynolds and colleagues are an amazing team, really just first in class, and we're very privileged to work with them. And I think the speakers who are working on the neuro and derm definitions made an irrefutable case that that work is foundational, both for developing treatment guidelines and also for biomarker development. Those definitions have to be granular and they have to be applied consistently in order to be able to find biomarkers or inform other autoimmune diseases. So to me, what came into focus for me today as an important challenge for us to address is that we need to, in parallel, have the ambition both to continue on this foundational definitional work and to amp things up in terms of looking for biomarkers and for shedding light on other autoimmune diseases. We can't do these sequentially. We have got to have the ambition to do them in parallel. Kerry showed us the explosion of numbers of patients on checkpoint inhibitors. That's just going to continue, and we owe it to those patients in order to leverage the great work we saw today along the lines of the strategy and the outline that Mark and Elad shared with us. So once again, fantastic program. Thanks. If you have questions or comments or want to follow up in any way, your go-to person here is Teilo. His email address is in the chat. So once again, thanks everybody. Really appreciate your contributions.